Many years ago, I worked at a hospital where I met a lady in her mid-30s. Let's call her Anne. A few years prior, Anne was perfectly healthy. She worked, she ran her own errands, but then she began to notice changes, like numbness in her arms and in her legs. Within a few years, her symptoms worsened, and now she needed a wheelchair to get from place to place. It turns out that she had multiple sclerosis, or MS. In the brain, we have these cells that produce a protective sheath called myelin to cover neuron axons. These axons are important for the communication that happens in our brain, and this myelin helps to protect the axon, much like the case that covers a wire. In MS, however, this casing is damaged, leaving the axons naked, and when these axons are damaged, it interrupts the signaling that occurs in the brain, causing the symptoms that people like Anne experience. Now, Anne had been healthy for most of her life. So why did the symptoms show up later in her life? Well, research shows that both genetics and the environment play a key role in MS. Many MS mutations are found in genes responsible for inflammation, and one of these genes is called the fractalkine receptor. This receptor is present in several different cell types in the brain that play key roles in producing that myelin. Now, the human population has a few different versions of this fractalkine receptor. There's the dominant form found in most people, and then several different mutant versions found in fewer people. Previous studies have found that individuals with the mutant receptor have worse MS symptoms, and also that their MS progresses faster than those who have the dominant version. So, naturally, scientists became very interested in studying this mutant fractalkine receptor. But how do we study a human gene in the lab? Well, they took the human mutant gene and they placed it into mouse DNA. So now we have these unique mice that produce the human mutant receptor instead of the mouse one. This is where I come in. The goal of my project is to find out whether this human mutant receptor affects normal development of the brain. To do this, I take juvenile mice brain and I look at the different cells, trying to see whether anything is different. So far, I've found that the mutant receptor affects the hippocampus, which plays a key role in memory and in learning. Mice with this mutation have much less myelin and the cells that produce it. This leads us to another key question. Are the axons developing normally? All of that is yet to be determined, but once we do, it may lead us to find early therapies such that people like Anne wouldn't need to wait until their symptoms appear, but potentially begin preventative treatment much earlier in life.